Genes are the building blocks of our bodies. We each have more than 20,000 genes that tell our bodies how to grow and function. Genes are made up of DNA, which is a type of code. This code is like letters in a book. When there are changes to the code, like spelling changes in a book, health and developmental conditions may occur. Examining our DNA or code is the key to finding a potential diagnosis. Chromosomes are the structures that package our DNA. All up, we have 46 chromosomes, 23 pairs, with a copy inherited from each parent. When all or parts of a chromosome are missing or duplicated, disability and health conditions may arise. There are many different types of tests to identify changes in our DNA. However, they can be broadly grouped into chromosome and genetic or genomic testing. Microarray testing looks for missing or extra parts in our chromosomes, or the chapters of the book, to try and find a reason for a genetic condition. This type of test has a 10 to 15% diagnosis success rate. Genetic and genomic testing looks for changes in the DNA or sentences of the book. A single gene test looks at only one gene to try and find if it is the cause of the genetic condition. A panel test is when more than one gene is looked at once. For example, a panel test may look at a group of genes that are known to cause epilepsy. This type of testing is done if a person has a pattern of features which may be linked to a particular gene or group of genes. This test is also useful if there is a family history of a particular condition. Genomic testing looks at a large number of genes or all our genes at once, providing a more detailed study of a person's genetic makeup. Genomic testing may include whole exome or whole genome sequencing. A trio genomic test is where samples from the child and both parents are also included. A whole exome sequencing test, also called an exome, looks at approximately 1 to 2% of a person's DNA, where most of the gene changes that cause genetic conditions will occur. A whole genome sequencing test, also known as a genome, looks at all of a person's DNA, or genetic code, to try and find the cause of the genetic condition. Exome and genome sequencing have up to a 50% chance of identifying a diagnosis, depending on the patient's observable characteristics. There are genetic counsellors throughout Australia who can provide further information. Don't feel like you're on your own. Join our Swan community and connect with parents and carers just like you. So don't wait a moment longer to become part of our wonderful Swan community. Contact us now.